Welcome to the video for chapter 4.3. So in the last video, we talked about lipids and carbohydrates, some of our macromolecules. In this lecture, we're going to talk about nucleic acids and proteins. So our second objectives for 4.3 are you're going to be able to describe the structure of DNA and explain how DNA stores information in its sequence of base pairs. You're going to be able to describe the structure and function of RNA, or the structure and functions of RNA, and you're going to be able to describe the fundamental structure of proteins. Okay, so let's talk about nucleic acids. Nucleic acids are composed of long chains of nucleotides. So the nucleotide is the monomer that makes up nucleic acids. So it doesn't matter if it's DNA or RNA, each nucleic acid is made up of these individual nucleotides. Now, a nucleotide has a very unique structure, but it's a consistent structure for both DNA and RNA. So there's three components to a nucleotide. The first is we have a phosphate group, this PO4 right here. We have a 5-carbon sugar. Okay, so 1, 2, 3, 4, 5 carbons. Okay, and then the last component of a nucleotide is what you're used to me calling nitrogenous bases. Okay, another name for a nitrogenous base is a nucleobase. Okay, so nucleobase is this ring structure here that has nitrogen in it. Okay, so one of the things you're going to have to be able to do for the next exam is you're going to have to tell me at least 3 differences between DNA and RNA. Okay, so let's talk about the first one. And this one is literally in the name of each of these. Okay, so DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Okay, what does this mean? So both DNA and RNA have a 5-carbon sugar, but they have a different 5-carbon sugar. So RNA has ribose. Okay, this is the ribose molecule. Okay, and then DNA has deoxyribose. So ribose and deoxyribose are almost the exact same thing, except on the second carbon here, ribose has an OH group, whereas deoxyribose has a hydrogen group. Okay, deoxy literally means lost the oxygen. Okay, so this is the first difference, is they have different 5-carbon sugars. Okay, another difference is their jobs. Okay, DNA is going to be our genetic blueprint. Okay, it contains all the information needed to help our cells make things and to run smoothly. Okay, while RNA, it participates in the transfer of information from our DNA, from our gene, to make a protein. Okay, well, we're going to talk about some of the structural components of DNA and RNA and how they differ in the next couple of slides. But one of the things that we need to keep in mind is DNA is more stable than RNA. This is another difference. Okay, RNA is the least stable, so meaning it's not going to last very long, while DNA is going to last considerably longer than RNA. Okay, so let's talk about the nucleobases. So one of the other differences between DNA and RNA is the type of nucleobases that they have. So in DNA contains the nucleobases adenine, which we represent with a capital A, thymine, which we represent with a capital T, guanine, which we represent with a capital G, and cytosine that we represent with a capital C. Okay, in DNA, these bases are going to pair up with each other. We call this base pairing. Okay, and they're always going to pair up with the same nucleobase. So adenine always base pairs with thymine, and guanine always base pairs with cytosine. Okay, so that's DNA. So RNA also has nucleobases. In fact, it shares some of the same nucleobases as DNA. Okay, so RNA has adenine, it has guanine, and it has cytosine. Okay, the difference between the nucleobases of RNA versus DNA is in RNA, we don't have thymine. So instead, we have uracil, which we represent with a capital U. Okay, so base pairing is the same in RNA, except we're going to have adenine base pairing with uracil because it doesn't have thymine. So guanine and cytosine still base pair in RNA as well. Okay, so we give names to the nucleobases. So we have the purines. Purines are always the bigger structure, even though it's the smallest word. This is how I remember this. Okay, so adenine and guanine are our purines. Okay, our pyrimidines are going to be the single ring structure. So this is going to be cytosine, thymine, and uracil, which is going to be our pyrimidines. Okay, so know the differences in bases and what bases base pair with each other. Okay, so let's talk about why knowing the structure of nucleotides and knowing the components of nucleotides is important. So we have HIV. HIV is the virus that causes AIDS. Okay, HIV is what we know as a RNA virus, so it's made up of just RNA. Okay, so HIV is pretty insidious. Okay, it's very terrible because one of the things that it does that other viruses don't is it integrates itself into your genome. So not only does it get into your T cells, it actually incorporates itself into your DNA. So every time a cell divides, we get that DNA being copied into the new cell. So this is why we can't just get rid of HIV. We'd have to literally go into our DNA and cut out that viral genome to get rid of it. And we haven't figured out a way to do this. Now, our HIV is an RNA virus. Okay, so what does this have to do with our DNA? Because we have DNA, not all viruses have DNA. Okay, so HIV is an RNA. It's actually what we call a retrovirus. So retroviruses do something kind of cool, also a little bit scary. HIV, because it's made of RNA, when the RNA gets into the cell, it's going to have the instructions to make a protein, an enzyme called reverse transcriptase. Okay, so this enzyme is going to do is it's going to take the viral RNA and it's going to reverse transcribe it, meaning it's going to make that RNA into DNA. Okay, and this is how that DNA can get integrated into your genome, because after it goes through reverse transcription, it's DNA. Okay, so why does this go with knowing what a nucleotide looks like? Okay, because we are going from RNA to DNA with reverse transcriptase, that means that we are building the DNA with bases that are found in the cell. Now, somebody got really smart. Okay, so how nucleotides are bound together is that they lose a water molecule, we go through a dehydration reaction, in order to pair them up. Okay, so the phosphate group is going to be attached to here. Okay, so to this 3' carbon, or third carbon in our base. And this is what's going to make those bonds that hold these nucleotides together. Now, the 3' OH, we'll talk about it in quite a few chapters in the future, but this is a very important OH group. Okay, both DNA and RNA have this 3' OH group on their 5 carbon sugar. Okay, so this is where we always add the next nucleotide. Now, somebody once we realized that HIV is a retrovirus and we're making DNA from an RNA viral genome, one of their first thoughts were, if we could interrupt the process of making DNA from the viral RNA, maybe we could stop HIV from infecting cells. So that's what they did. So they made up this molecule, okay? So it's called AZT. I can't remember what its medicine name is, but we'll just call it AZT. So AZT has this structure. So it has what looks like a nucleobase. We have a 5 carbon sugar, and we have 3 phosphate groups. Now, what's really great about this and what's really ingenious about this is this right here. So on a regular sugar in DNA and RNA, this would be an OH group. Now by putting an N3 group here, we are going to terminate or end the DNA molecule. We're going to stop the chain. So how we add this guy to our sequence of bases is we are going to break the bond in between this phosphate and this phosphate. Okay, so breaking this bond is going to provide energy, because remember, breaking bonds always gives us energy. Okay, and then this is going to link up with this OH group, and we're going to bind it, and we're going to add this to our DNA sequence. Now, because this is added, this is going to stop that reverse transcription, because there's nothing to add the next base to, the next nucleotide. Okay, unfortunately for us, HIV, this worked for quite a long time actually, but HIV has since evolved and adapted so that this kind of medication doesn't work anymore. It's able to get rid of it and continue transcribing its RNA to DNA with reverse transcriptase. Now, if you remember from biology, our AZT molecule from the previous slide looks very familiar to another really important molecule in biology, and that's ATP. So we're going to have our nitrogenous base, done. In this case, in ATP, it's adenine. Okay, we have our 5-carbon ribose sugar, just like in RNA, and we're going to have these 3 phosphate groups. Okay, so we have our triphosphate. Okay, we use breaking the bonds between these phosphates to power reactions. So in the previous one, we broke it between those first 2 phosphates. Okay, on ATP, we do it between the beta and gamma phosphate, and that's going to provide enough energy to power a lot of reactions. Okay, this is why the ATP molecule is so important. Okay, so let's talk about some more differences between DNA and RNA. And this time, let's talk about some structural differences. Okay, one of the easiest ones to remember is that DNA is a double helix, which means it's double-stranded, while RNA is single-stranded. Okay, now there are multiple types of RNA, and we'll discuss a couple here in a second. What we need to remember is when we're talking about the single-stranded version, we're talking about mRNA. Okay, so all chromosomes, so all of our genes, are made of this double-stranded DNA. Sometimes we refer to the double-stranded as a duplex. Okay, so in this molecule, we have the 2 strands. Okay, they're anti-parallel, meaning they're going in opposite directions. Okay, they kind of look like a winding staircase. Okay, and the bases, those adenine and thymines and guanines and cytosines, are going to make the rungs of the ladder. Okay, or the steps on the stairs. Okay, the turns are caused by hydrogen bonds. Hydrogen bonds are important in DNA. Okay, so DNA a double helix, while RNA is going to be a single strand, which allows mRNA to be a messenger. So mRNA is the molecule that takes the information stored in our DNA and then uses it to make proteins. So it's kind of like the middleman, if you remember our central dogma of biology. Our central dogma of biology says that DNA is always going to go to RNA, and then RNA is going to go to make proteins. Okay, so the mRNA is going to take that information from the DNA and go to the ribosome to start making proteins. Okay, we already talked about this, double-stranded versus single-stranded, and the bases are different. So you guys know everything on this slide already. Okay, so one of the things that we need to talk about are the different types of RNA. So there's 3 main types of RNA. There are a couple different ones outside of these, but these are our 3 most common ones. Okay, so RNA can come in 3 types. We've talked a little bit about mRNA, our messenger RNA. This is going to be transcribed from a gene and used to make proteins. So the other 2 that we talk about a lot are rRNA, which means ribosomal RNA. Okay, so RNA is part of that ribosome, the thing that makes proteins, and tRNA is what we call the transfer RNA. I like to think of the T as in taxi, because what tRNA does is it carries amino acids to the mRNA and the ribosome complex so a protein can get made. Okay, so we talked about mRNA and how it's single-stranded. tRNA has a different shape, so it's going to have this kind of T-shape. Okay, so if you remember back from biology, we talked about this. So we have our sequence of bases making up our tRNA. What's going to happen is this tRNA is going to bind an amino acid up here, okay, and then it's going to bind to the mRNA, so it knows where in the sequence of amino acids in the protein to place that amino acid. And it does that by what is called the anticodon. Okay, so each base, if you remember, is represented by, we use these bases in sets of 3 to represent each amino acid. We call that a codon. Okay, so the anticodon matches the codons, so the sets of 3, and the mRNA. And this is how the tRNA knows which amino acid to add to the protein next. Okay, there's quite a few different viruses, such as the influenza virus, SARS-CoV-2, so the COVID virus, and HIV that have RNA genomes. Okay, because remember, this is something that's different, that DNA can have both an RNA or DNA genome. So when we talk about mRNA, we have to talk about one of the biggest stories in the last 5 years, and that's the fact that scientists developed a vaccine that uses mRNA to vaccinate us against COVID. Okay, so let's talk about this real quick. So we have our COVID virus. So we looked in one of the previous lectures, we looked at the electron micrograph of COVID, so we could see the little spike proteins on the side. Okay, this spike protein is unique to the COVID virus. No other virus really has the spike protein. Okay, so what we did as scientists is we sequenced the genome of the virus, and we looked at the DNA sequences, and we found the specific piece of that DNA sequence that codes for the spike protein, and we made the complementary mRNA that goes along and matches that DNA of that spike protein. Okay, so we took the mRNA that codes for that spike protein and we put it into a lipid nanoparticle. Okay, so this is the vaccine, just this strand of mRNA in a lipid nanoparticle. Okay, so this is going to get administered into your upper arm, into the muscle tissue. What's going to happen is this lipid nanoparticle is going to kind of float around through your tissues until it comes into contact with a muscle cell. And the plasma or cell membrane, which is made of lipids, is going to allow this lipid nanoparticle to join it. And what's going to happen is that's going to open the lipid nanoparticle up and release the mRNA into the inside of the cell, into the cytoplasm. Okay, cytoplasm is where mRNA gets used to make proteins. So your molecular machinery in your muscle cell is going to say, hey, mRNA, we need to use this and make the proteins that it makes. So our body's going to make a ton of these spike proteins. Okay, they're going to get shipped outside of the cell into the bloodstream. And what's going to happen is in the bloodstream, we're going to have our T cells, our immune system molecules, that are going to find these spike proteins. And it's going to be like, oh, hey, this doesn't belong. And that's going to trigger an immune response. So our immune system starts to build antibodies, which is these little Y-shaped guys. And these antibodies are going to be specific for the spike protein. Okay, so this is a way to train your immune system to recognize the COVID-19 virus without actually getting COVID-19. Because the spike protein, when it's made from the RNA and it's using this vaccine, this protein is harmless. It doesn't cause disease. If anybody ever tells you that they got COVID from the COVID vaccine, they are lying their pants off. You cannot get COVID from the mRNA vaccine. It's not the virus. It's just the mRNA code for the protein. Okay, so this is a way that we can train our bodies to recognize COVID without actually getting it and having those harmful side effects, those harmful signs and symptoms. Okay, it's actually really quite an elegant system. We've actually known about mRNA vaccines for the last 30 years, and we've tried to develop them for a bunch of different diseases. In fact, a bunch of different COVID or coronaviruses like COVID is. But really the breakthrough with the mRNA vaccine and what made it so effective is this lipid nanoparticle. This was the last piece that we needed to make the delivery system to make sure that the mRNA gets into your cells before it becomes degraded or broken down. Okay, so that is nucleic acids. We are going to now go through and talk about proteins. Proteins are probably the most diverse type of biomolecules because proteins are everywhere in a living organism. They do so many things. In fact, you guys probably remember in biology that I used to say for proteins, shape denotes function. Okay, each protein shape is unique, and that protein is going to have one job, which is why we have so many different types of proteins. We have proteins on the outside of the cell that are going to communicate with the cells next to them. Okay, so these can serve as both receptors. We can have proteins in the membrane that act as transporters that are going to bring things in and out of the cell that can't just cross the plasma membrane. Proteins are going to act as what we call catalysts, so things that speed up reactions in the body. Okay, we're going to have proteins that provide structure. We're going to have different proteins that help the cell move. Okay, but all proteins, no matter their shape and their job, all have a unique monomer. Okay, so proteins are made of amino acids. Okay, and these protein monomers have 4 parts. Okay, so we have a central carbon. Okay, attached to this carbon, because carbon can make 4 bonds, is an amino group. So amino group is an NH3 plus group. It has a single hydrogen with one of the bonds. Okay, we have this carboxyl group. So this is a carbon double bonded to oxygen and single bonded to a O minus. Okay, but what really makes each amino acid different is this R group. Okay, every amino acid, and we have 20 of them, have a different R group, so different side chain. This is the important part of the amino acid. This is the part that does the chemistry. Okay, so we have 20 different amino acids. This is a great chart. And no, you do not have to memorize it like you had to do in Biology 201. So you can kind of just see as we go through here, the different R groups or side chains that our amino acid can have. So we have some, let me find it. We have lysine. This one's negatively charged. Okay, we have arginine, which is positively charged. We have glutamic acid, that's negatively charged. We have, where is it? Gotta find it. Give me just a second. We have phenylalanine, that is pretty much nonpolar. We have some that are acidic. I cannot never remember which ones. Oh, the ones that say acid. These ones are acidic. Okay, we have some that contain sulfur, like cysteine and methionine. Okay, these are going to come in handy when we talk about the different levels of protein structure. Okay, but one thing I want you to know is what an essential amino acid is. So whenever we talk about essential things, so essential amino acids, essential fatty acids, anytime we use the term essential in biology, that means that we cannot make this thing. We have to get it from our diet. So like isoleucine, leucine, valine, these are all essential amino acids. We have to get these from our diet in order to make proteins. Okay, the last thing we're going to talk about in this lecture are the 4 levels of protein structure. Okay, so how do proteins get their unique shapes? Okay, so proteins, when they're functional, are going to have a unique 3D shape. How do we get from a sequence of amino acids to this 3D shape? And that is all has to do with our levels of structure. So our first level of protein structure is the primary structure. Okay, the primary structure is literally the linear sequence of amino acids. Okay, so we're going to start with a serine, the next one's going to be a phenylalanine, the next is going to be an aspartate, next is going to be methionine. Okay, this is the sequence of amino acids in this small little peptide here. Okay, the amino acids are held together with what we call a peptide bond or a peptide linkage. Okay, we're going to take the OH off the carboxyl group and one of the H's off the amine group, and this is going to make water and form a bond. Remember, all these are combined by a dehydration reaction. Okay, so we find the peptide bonds in the first level protein structure, which is the primary structure. You need to know that. You'll be tested on that. Okay, so sometimes we have students that get a little bit confused between peptide and protein. What do they mean? For a lot of times, peptide and protein mean the same thing. So peptide means a single sequence of amino acids. And a lot of times we can say that for a protein as well. A protein is just one linear sequence of amino acids. In a few levels of structure, I'll tell you the other meaning for protein and why sometimes isn't the same as a peptide, but we'll get there. So protein sequences or peptide sequences are chains that can be hundreds into thousands of amino acids long. So it all depends on what that protein needs to do and what shape it needs. Okay, our second level of structure is what we call secondary structure. Very easy to remember. Okay, so, well, primary structure is literally our sequence of amino acids. Our secondary structure is when we start to fold, where we start to get that shape that's going to give our protein its function. Now, in secondary structure, we call this the local folding. Okay, so we're going to start to fold little pieces of the protein. Okay, in secondary structure, we only make two types of structures. We make the alpha helix, which is this kind of curly fry shaped guy, or we make a beta pleated sheet. So kind of in these pictures, kind of looks like an unfolded map, like you'd find in the card jockey box. Okay, so these are the only things that are made in this level of protein structure. Now, if you remember from Biology 201, I talked about mad cow disease, so bovine spongiform encephalopathy. Okay, so if you remember, mad cow disease is caused by prions. Okay, so these are misfolded proteins that cause healthy proteins to also misfold. Okay, so we talked about how prions in mad cow disease affect the next level of structure, that tertiary structure. But what we didn't really talk about in Biology 201 is that the fact that prions affect the secondary structure. In fact, in mad cow disease, any alpha helix in mad cow disease is literally going to be misfolded. So what it's going to do is it's going to cause our healthy protein that has these alpha helices in, all these alpha helices are going to be converted to beta pleated sheets and not be the same. So this is one of the ways that prions can cause our healthy proteins to misfold. We literally convert the alpha helix to a beta pleated sheet, which is going to change its shape, which means it's going to change its function. Okay, so level 3 of protein folding or protein structure is the tertiary structure. Tertiary means 3. Okay, the tertiary structure is when we get the unique 3D shape of a protein. This is when we completely stop folding. Okay, so we're going to go from secondary structure with our alpha helices and beta pleated sheets, and then we're going to do our last level of folding to make the functional 3D structure of our protein so it can do its job. Okay, we have different ways that it can fold. We can have hydrogen bonds between like oxygens and hydrogens, nitrogens and hydrogens, sulfurs and nitrogens. We can have hydrophobic interactions. So we have some hydrophobic or non-polar R groups on amino acids. So they don't want to be in water, so the protein's going to fold to create a pocket for them inside the protein where they don't have to interact with water. We can have ionic interactions. We have all those that were positively and negatively charged. We can create an ionic bond between these two R groups that's going to help the protein fold. Okay, and we can also if we have one amino acid close to another amino acid with sulfur in it, okay, we can make what's called a disulfide bond or a disulfide bridge, because the sulfurs will actually bind to each other to help the folding. Okay, so in a lot of proteins, level 3, so tertiary structure, is the last level of structure that they need. If we have a protein that is just one single amino acid sequence, tertiary structure is what is all that there is for that protein. Once it has its 3-day shape, it can then go and be used. Okay, one of my favorite and one of Dr. Burroughs' favorite proteins is what we call GFP. So GFP stands for green fluorescent protein. It has been isolated from jellyfish. And what it does is it's the thing that makes some jellyfish glow in the dark. Okay, so we can take GFP and we can attach it to other proteins and inject it into frogs or mice or other animal. Fish, zebrafish also have GFP, and we can make it glow in the dark, which is kind of cool. Okay, but GFP is an example of a protein that stops at the tertiary structure. Now, remember I said tertiary structure is the main structure that's interrupted by prions in mad cow disease. Okay, so our misfolded protein is going to not have the correct 3D shape, so the correct tertiary structure, in order to do the job that it was originally built for. Now, once a healthy protein misfolds because of prions, what's going to happen is that they're going to be more inclined to aggregate or clump together. And what happens is in mad cow disease, these misfolded proteins are going to clump together, and it's actually going to start to create holes, like a sponge, hence bovine spongiform encephalopathy, literally start to create holes in the cow's brain. Okay, so you probably remember this one. This one is a slide from my Biology 201 slideshow. Okay, so this is the healthy brain tissue of a cow without mad cow disease, while this one is a micrograph of the brain tissue of a cow that has mad cow disease. Okay, so we can see everything's pretty uniform. There's no holes up in this top one, but in this one, we can literally see the holes like a sponge in this cow's brain. Okay, so this is going to affect the nervous system of the cow. It's going to start to have seizures. It's going to literally go mad. They're going to shuffle around, kind of like have a zombie-like shuffle for a cow. They're going to become insomniacs. In fact, a lot of things that get a prion disease are going to be affected by insomnia, meaning they're literally going to stay awake for the rest of their lives and will literally die from lack of sleep. Okay. So this is, and mad cow disease is passed to other cows when they accidentally get the nervous system tissue or brain tissue of an infected cow and ingest it, so they eat it. Okay, we have a lot of regulations set by the FDA and the agricultural agency for the government. I can't ever FDSA, I think is what it's called. We have a lot of regulations now that allow us, well, that don't allow us, that tell us that we can't feed cow parts to cows. Okay, so we don't really see a lot of mad cow disease here in the US. It was more prevalently seen in Canada and in the UK, where there wasn't such strict feed laws for livestock. Okay, so this is the last slide. We're going to talk about the fourth level of protein structure, which is called the quaternary structure. Now, a lot of proteins are done at tertiary structure if we only need one strand of amino acids to make it. Now, there are a lot of things like the heme in your blood or the antibodies that attack antigens in your bloodstream that are part of your immune system that are going to have quaternary structure. So what quaternary structure does is that we're going to have more than one amino acid chain going to make up the protein. So in the case of an antibody, we literally have 4 chains. We have 2 heavy chains and 2 light chains that are going to complex together to make our antibody. So we're going to get this characteristic Y shape. And these parts right here are going to be what we call the antigen binding sites. Okay, so this is where your antibody is going to bind to the thing that it's specific for. Okay, so we talked about in class that red blood cells have antigens. So if you have the B blood type, you have the B antigen on the outside of your blood cells. If you have the A blood type, you have the A antigens. If you have AB, you have both A and B. And if you have the O blood type, you don't have any antigens at all, which is why it can be a donor. So if you are someone that has B type blood and you're given A blood, you literally have antibodies called anti-A's in your bloodstream that are going to target that red blood cell with the A antigen and recognize it as not being part of you, and it's going to attack it and get rid of it. This is why if you get the wrong blood in a blood transfusion, that you get really sick, because this is going to elicit an immune response pretty quickly. Okay, in the terms of your red blood cells, in your red blood cells you have a complex called heme. Your heme is going to have, I think it's 4, it might be 8, I can never remember. So it's going to have at least 4 peptide sequences, so amino acid sequences, that come together to make this protein complex with iron. It's going to be what carries the oxygen around in your blood for your red blood cells. Okay, so quaternary structure is going to be more than one peptide chain combined to make a protein. So proteins can be single amino acid chains, or they can have multiple amino acid chains, whereas a peptide is always going to be a single one. Okay, so that finishes 4.3. We went through the differences between DNA and RNA and what makes up a nucleotide. We went through proteins and the fact that they're made up of different amino acids, and we talked about the protein levels of structure. I would definitely know the levels of structure for the exam, as well as the main differences between DNA and RNA. We will see you in 4.4.